Hey everyone. I know it's been a while since I've made you watch a video lecture for homework, but we're back to it with membrane structure. So let's get started thinking about it. So the first thing we're going to talk about is, I feel like there's this word or phrase, I guess is more appropriate, the phrase fluid mosaic that always gets referenced to when we're talking about the cell membrane. And I want to just take a minute and break down why we use this this phrase when we're talking about the cell membrane and what it means. So the fluid mosaic term is actually more of a model. So there were different, before we knew sort of how the cell membrane functioned and the way it behaved, there were different ideas about how it did that. And eventually now we know that this fluid mosaic model is the closest thing that we've figured out for how the cell membrane functions. So let's take this word by word and think about the word fluid and the word mosaic. So like you might expect, the word fluid means that the cell membrane it behaves like a liquid. So people have actually done studies about the fluidity of the cell membrane, and they found that it has the viscosity of something like olive oil. So we know that or at least at this point, you should know that the cell membrane is made of these molecules called phospholipids. So that's all of these little red spheres with the yellow tails here. And so we have this sea of phospholipids, and embedded within those phospholipid layers, we have all kinds of different molecules. So you can see here, some are labeled. We've got proteins that can be either transmembrane proteins, so that means they span both layers of the phospholipid bilayer. They can be embedded in just one of the layers. So you can see this layer here is just embedded on the cytosol or the cytoplasm side, whereas maybe a protein like this could be just on the outside or the extracellular space of the cell. We've also got these chains of different kinds of carbohydrate containing molecules. So these are called glycoproteins. And so usually they have some sort of protein bit that helps keep them anchored in the membrane, but then they've got these extra molecules that are sugars that are hanging off, and that helps with cells communicating with one another. It helps to identify the cell, like to tell other cells what kind of cell it is. And then we've also got these cholesterol molecules that are studded inside of the hydrophobic tail portion of the membrane. So we've got all of these different kinds of molecules that are hanging out inside of the phospholipid bilayer, and these all refer to the mosaic part. So if you've ever looked at a mosaic, you know it's these pieces of colored like tile or stones that are embedded in like a concrete or cement. So with the cell membrane, the phospholipids are going to be like the cement, and then all of the colored stones are going to be these proteins and cholesterol and glycoproteins and all of these other molecules that are sort of stuck inside of it. So that refers to the mosaic, and then the fact that all of these things, they're not fixed in place. So like this membrane protein right here, maybe we're looking at a snapshot of it right here, but maybe in 10 minutes it's going to be over here, because all of these things are mobile and can be moving around because of the fluidity of the membrane. So that's what we're talking about when we say the fluid mosaic. It's just the fact that the membrane has these two characteristics. It's fluid, or it's liquid, and it's also a mosaic of different kinds of molecules. Okay, so I just spent a little bit making a big deal about how the membrane is liquid. And so I want to take a minute and have us think about how liquid or how fluid is the membrane. It's not a fixed quantity where it's always the viscosity of olive oil, right? So some membranes can be a little bit more fluid. Some membranes are a little bit less fluid. So think about how we can test or measure quantitatively how fluid a membrane protein is, or a membrane itself. Those are kind of one and the same. If we're tracking a membrane protein, we're also sort of by extension testing how fluid the membrane itself is. So here are a couple hints. One, we can use tags to fluorescently label macromolecules. So that means we can make these macromolecules fluoresce or glow with specific colors. We can also use lasers to bleach these fluorescently labeled molecules, which just means that we're going to stop their fluorescence. We're going to make it so they don't glow anymore. And if we were in class, I would make you talk with your table before answering this question for you. But instead, since this is a video, I will ask you to pause and just, I mean, don't take forever, but think about this for a minute. How could you use technologies like the ones we just listed to measure how fluid a membrane is? Okay. Okay, so now I'll tell you the answer in the next couple slides. So one early experiment that people did was to use two different kinds of cells and label them with different colors of fluorescently labeled proteins. So here we're showing, it's really hard to label within one given cell. You kind of have to keep it the same color. It's really hard to have different kinds of fluorescently labeled colors on one particular cell. So instead, they took two different cells. Here we have a mouse cell and here we have a human cell. And the mouse cell is labeled in red and the human cell is labeled in green. And they fused them so that now we have a giant cell that's half red and half green. And they waited for, here it says 40 minutes, and found that after 40 minutes, all of these red and green tags had spread and were not where they started. We started with this sort of perfect, nice split between the red and the green. And after 40 minutes, you can see that the colored fluorescent molecules are just all over the place and they're totally mixed in with one another. So that tells us that all of the membrane molecules and proteins that started off down here, now half of them are up here and half the ones that started up on the top are now down at the bottom. So this shows us that the membrane proteins are fluid and mobile. But it doesn't let us get a very quantitative number. We can say here they're perfectly mixed, but what if after 30 minutes they were sort of like mostly mixed, right? It's a little bit hard to quantify something like that. So instead, today, the kinds of experiments that we use to measure how fluid a membrane is this other kind of experiment that uses the lasers that I was mentioning. So we call this fluorescence recovery after photo bleaching, and that's kind of a mouthful. So most people call this FRAP. These are called FRAP experiments. And the way it works is we have a cell, and usually they're showing this one as obviously cells don't naturally have fluorescent proteins on their surfaces. So the first step is going to be to label some sort of fluorescent dye or some sort of fluorescent tag to cover the entire surface of the membrane with these fluorescent molecules. So you come in with this laser, and the laser is so it's so high energy that it breaks or alters the interactions or the bonds within these fluorescent molecules, and it makes it so that they're not able to fluoresce anymore. So this region is permanently like not fluorescing anymore. It's just kind of done. And then so what you would expect to see is that if the membrane is fluid, that these other molecules that are still fluorescing will come in and fill the space that was left behind with this photo bleached spot. And obviously these bleached spots are also going to move and sort of take over the rest of the membrane as well. But eventually you'll even out, and then there will be an even amount of fluorescence that is taken over where this photo bleach spot was. And so when you graph this, the results of an experiment like this, you end up with a curve that has this kind of shape. So we're looking at intensity of fluorescence specifically within this region where we bleached it. So you can see that before the laser came in, we've got, this will be our 100% fluorescence mark. It's always, it's relative units. So we're always measuring it relative to before the laser came in. And then once the laser bleaches that area, you get this immediately very sharp decrease of fluorescence down to 0. And that's what we're looking at here, where right after the laser hits, there's no fluorescence left. And then the next part is this recovery aspect. So as the membrane molecules are moving around and filling in that spot, we see this gradual recovery of of the fluorescence in that region. So eventually you'll notice that this end point, so here it's fully recovered and it's sort of just leveling off here. If the graph continued, you'd see that it would just go like this. So if you compare it to before the laser hit, you'll see that it's not 100%. And that's because we did permanently remove some of the fluorescent molecules. So even if they perfectly redistribute and these other molecules come in, on the whole, we've got less fluorescence in this cell after the photobleaching than we did before. So you're never going to get back up to 100%, but you can still get up to 80 or 90. And so that's what we're looking at here at the end. That's why it doesn't go all the way back up. And then the steepness of this curve lets us say how fluid the membrane is. So if you get a membrane that's super, super fluid, really, really mobile, then maybe the recovery curve would look something more like this. You get a steeper, faster recovery curve. And if the membrane is really, really immobile and slow, it's not going to get more immobile than this, but this is showing a pretty immobile one. So it can either go like this, if you're following my pointer, or it can go like this. And that'll tell you quantitatively how mobile or how fluid the membrane is. So just to give you a more realistic picture, these are all diagrams of what these results actually look like. Here it's showing a real cell, and you can see that you have to like trace out using computer software this region of interest where the bleach is going to happen. And then after the bleach, you get this big black hole of non-labeled membrane. And then after the recovery comes back in, you can see that we've got an even distribution of the rest of the fluorescence in the cell. And you can see, like I was saying before, it doesn't get back to full fluorescence. You can see this cell is brighter than this cell, so we don't get full recovery, but you don't have this black hole sitting there either, because all of the extra fluorescent molecules that are hanging out here are going to fill in this space. So this is showing another one of the graphs, and you can see this recovery curve is a little bit steeper. So instead of having a curve that looks like this, we have this curve that bounces back pretty quickly. And so the steepness of this curve is going to tell us how quickly our recovery is happening, and therefore by proxy how fluid our membrane is. Okay, so our next slide has to do with another factor that has an effect on membrane fluidity, and this next factor is temperature. So membrane fluidity is going to depend on temperature. And I think this should probably be pretty intuitive to people. If you've ever had butter that you've kept in the fridge versus out on the counter on a hot day, you'll know that the butter is a lot more liquid if you keep it outside in warm temperatures versus inside of the cold fridge. The same thing is true, like if you make a dressing with olive oil, and you put the dressing in the fridge, you know, the olive oil can get kind of like chunky and gross. So those two substances, butter and olive oil, they're both lipids, just like a phospholipid. So phospholipids in the membrane are going to behave in similar ways. So here you can see what's called the melting curve of a phospholipid bilayer. And maybe you'll notice it's got this sigmoidal, it's got this S-shaped appearance to it. It's got this interesting inflection right here where it goes up really steeply and then it doesn't change very much, and it also doesn't change very much over here. So that's the characteristic curve for a melting curve for a membrane. And so what we're transitioning between here, what you're looking at is on the top side, or on the sort of this upper region, that's when the membrane is more fluid. So this is when I was talking about it being more like olive oil, really mobile. That's what we're seeing up here. And then down here, we're thinking of it more when it's like a gel or sort of more rigid or ordered. So when we transition between these two states, you'll notice that it goes from being more solid to more liquid as you increase the temperature. And then secondly, there's this point where we get a sort of sharp transition between these two. So it's not a linear, like gradual transition. There is a point at which we go like between this more fluid-like and more solid-like state. And this midpoint between where this transition is happening is called the TM. So that's the temperature at which the membrane is transitioning between this more solid-like state and this more liquid-like state. So membrane fluidity depends on temperature as well, and we're talking about more warmer temperatures, meaning more fluid-like characteristics, cooler temperatures, meaning more solid-like characteristics. Okay, so membrane fluidity and temperature, and there are just a couple other factors that we're going to go through in terms of other things that affect membrane fluidity. One of those characteristics is going to be the degree of saturation versus unsaturation in the membrane. And you should have an idea about this, what this means from the activity that we did in class. And then also the molecule cholesterol and the effect that it has on membrane fluidity. So we'll talk about saturation first. Okay, so remember that unsaturated phospholipid tails have a double bond within them, whereas saturated phospholipid tails have no double bond and they're more straight. So because these phospholipid tails with at least one double bond have these bends, they are sort of, it's almost, I think of it as being sort of more like they need more personal space. Like you can't get too close with them because it's almost like if you're standing there and you put your arm out to the side, like you're basically telling somebody like, yo, don't get too close to me. I need some room. They're socially distancing over here, whereas here they're just packed in like sardines. So these unsaturated, and this is a sort of a side note, but this word saturation, it's essentially referring to whether or not the tail is saturated with hydrogens. So if you think about there being a double bond, this would be easier to explain if I had a diagram right here. If there's a double bond, that takes up one of the spaces that the carbon has for a bonding partner. So normally a carbon has 4 spots where things can bind to it. But if it's got a double bond as one of those spots, then it's only able to bind to 3 things. So whereas all of these tails have 4, whereas these tails, because of the way that the tails are set up, each carbon has 2 hydrogens. Some of these tails would only have 1 hydrogen. So they're unsaturated with hydrogens, whereas these ones are saturated. But because of this, at least 1 degree of unsaturation is going to cause these kinks to form in the membrane, and then it's going to sort of force the molecules to be more spread out. And because they're not as tightly clustered to one another, that's going to give the membrane a more fluid composition. So when you think about it, what's the only thing that's really holding a membrane together is the fact that these hydrophobic tails don't want to be exposed to water, whereas these hydrophilic heads do. Like there's no bonds that are connecting this phospholipid to this phospholipid. It's just those interactions of hydrophobic attractions down here and hydrophilic attractions right here. And so because that's the only thing that's holding the membrane together, you get a decrease in those attractions when the molecules are spread out like this, and that causes the membrane to be more fluid. Okay, and then last but not least, it's not the last slide, it's just our last effect on fluidity slide, so don't get your hopes up too high. We have cholesterol. So cholesterol is this steroid molecule. It's a steroid because it's got this ring structure, and it's, for the vast majority of it, it's hydrophobic. So the way that these diagrams work, I'm not sure if you covered this in chemistry. If you ever take OChem in college, that's when you'll learn all about this. Basically, wherever a corner on these diagrams happen is a carbon. So like this is a carbon right here, 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 etc. And everything that's not shown, like here they show an O, but all of these carbons are filled in to the max with hydrogens. So all of this is a bunch of carbons and hydrogens, which we learned that kind of bond is nonpolar. So the vast majority of cholesterol is nonpolar. And then we have this tiny little polar head group with the OH right at the very end. So when you're orienting cholesterol within a membrane, you should be able to sort of recognize that these 4 rings right here are referring to this bit of the cholesterol. And this tiny little bit that's sticking out with the hydrophobic heads is going to be this one polar head group. So that's why cholesterol sits in the membrane the way it does. And the last, one of the last questions that I asked you in the membrane structure exploration Google slide thing was what effect you thought cholesterol would have on membrane fluidity. And I'm curious to see what people's responses were. I think that people think of 2 possible answers for this. So one possible answer is that cholesterol sort of acts like a kink in the membrane, where it's sort of pushing these two molecules apart and decreasing the interactions between them, and therefore making the membrane more fluid. So that's one way to think about it, that cholesterol essentially acts like an unsaturated tail. The other possibility that I think comes intuitively to people is that cholesterol actually packs the membrane tails more tightly together. So it's sort of acting like a saturated tail, where now they're packed even more like sardines than they already were, because all of these cholesterols are just sort of like forcing their way in there. And so if that was the case, then cholesterol would make the membrane less fluid than it already was. So what's the right answer? The right answer is actually both. Both of those things can be true. And how it's going to work is it's going to depend on on whether the membrane is above or below that melting temperature that I mentioned earlier. So remember, down here, we're talking about the membrane being more solid, more like a gel, more rigid. And so when the temperature is low, cholesterol is going to have the effect of increasing membrane fluidity. So acting more like an unsaturated tail, and sort of forcing the phospholipid molecules to spread out a little bit more and increase the fluidity. But at higher temperatures, where we're more into the fluid-like state, obviously, if you think about, like, if you left butter out in the sun, it gets too liquid, right? It's just like it doesn't have structural integrity anymore. And the same thing could happen to our membranes at really high temperatures. So at really high temperatures, where we're above the melting temperature, cholesterol is going to actually decrease the fluidity of the membrane and bring it down a bit. So it's going to act more like a saturated tail and increase the structure and the rigidity of the membrane. So cholesterol sort of modulates the fluidity of the membrane above and below the melting temperature in opposite ways. So it basically keeps us closer to a happy medium. In which direction it's pulling fluidity is going to depend on whether we're above or below this melting temperature, which I think is a super cool thing. Okay, our last couple slides, now I can say we're on our last few, have to do with sort of properties of the membrane, and functionally what we're thinking about. Like, why do we care about membrane fluidity? Maybe I should have led with this. Oh, and one more thing before we get there. This is a quick point I just want to make. We talk about the phospholipid bilayer, and I think it's easy to just assume that both layers, so like the outside layer that's facing the extracellular space or the extracellular matrix, and the inside layer that's facing the cytosol, it's easy to think of them as being exactly the same, like two pieces of bread that are making a sandwich. But they're not actually symmetrical. There's very different chemical properties that the outside phospholipid layers have that the inside phospholipid layers don't. So it's very easy when we say the membrane is fluid. What we mean is the outer phospholipids can switch places with other outside phospholipids, and the same thing for the inside phospholipids. But so this lateral diffusion happens very quickly, but there's very little diffusion or switching of an outside phospholipid onto the inside, or vice versa. So there are actually specific enzymes called flippase and floppase. There's one called scramblase too, that are responsible for changing the chemical properties of an outside one to make it an inside one, and vice versa. So transverse diffusion or flip-flopping doesn't happen very often. I'm not going to get into the chemical names, but yeah, there's very specific kinds of phospholipids on the outside and on the inside. We'll leave it at that. Okay, so thinking about fluidity of a cell membrane, like, why does that matter? It matters because the fluidity of a membrane is going to be really tightly connected to the permeability of a membrane. And when we're thinking about a cell membrane, the word that people use, the same way they use the word fluid mosaic when they're talking about its structure, they use the word selectively permeable when they're talking about its when they're talking about what kinds of molecules can move across it. So cell membranes are selectively permeable, and in the same way that maybe like specific colleges can be highly selective, that means they're really choosy about who they're letting in and who they're not. Membranes are selectively permeable too. So not just any molecule can move across the cell membrane. So when we're looking at what kinds of molecules are able to get across, there are sort of two characteristics that we have to take into account. One is the size of a molecule. So because this phospholipid bilayer, regardless of whether there are unsaturated tails or not, it's kind of tightly packed together. It's got it's a layer that the molecules have to cross, and it's just physically easier for smaller molecules to get across the layer than bigger molecules. So if you're looking at this large glucose molecule, like there's just no way it's going to force itself through this phospholipid bilayer. But if you're thinking about smaller molecules, like maybe this nitrogen, you can imagine it'd be easier for it to kind of sneak its way through. So smaller molecules are going to have an easier time getting across. The other factor that we have to take into consideration is whether the molecule is nonpolar, polar, or charged. So because there's this dense sort of region of these hydrophobic or nonpolar tails, this is going to be the sticking point for molecules as they're trying to move across the membrane. So uncharged, nonpolar molecules like oxygen, carbon dioxide, nitrogen, these are going to have the easiest time because as they get through into this hydrophobic region, they feel right at home. They're like, oh yeah, nonpolar, I'm nonpolar too, we can hang out. And so they'll be able to easily diffuse, and we'll get into diffusion in a little bit. They'll be able to cross the membrane and get to the other side with relative ease. However, if you think about a charged molecule like chloride or calcium, as it's trying to move across, it's not going to be able to interact with these hydrophobic tails at all. Like the hydrophobic tails are going to say, this is a hydrophobic region only. You've got a charge. We're not about that. So they're not going to be able to diffuse across the membrane, even though they're really small. And then polar molecules, it's kind of like an in-between. If they're really polar, then they're not going to be able to go, especially if they're big and polar, then they're not going to be able to go. Water is able to get across the membrane like a little bit because it's so small, even though it's polar, but it actually needs a lot of channels to help it get into the cell. So we're going to say small, uncharged, you're good to go to get across the membrane without any help. Large and polar molecules, unable to cross the membrane unassisted, they're going to need special transporter proteins to help them get across. I think, oh yeah, these need a special channel or a transporter to move across the membrane. And I believe that is our last slide. Oh wait, no, I lied. This is the last one, I promise. So like I said, like I was mentioning earlier, the reason that we're thinking about, or one of the reasons that we're thinking about membrane fluidity is that it's related to permeability. So generally speaking, the more fluid a membrane is, the more permeable it's going to be to molecules. So we were talking about how fluid membranes, be it because of temperature or unsaturated tails, there's more space or more gaps in between each phospholipid, and that leaves more room for molecules to move through. It's easier for them to cross. This is going to be most important for molecules that are sort of like it's in between. So I was talking about how water is polar, but it's so small that it can sort of cross the membrane sometimes. So it's more important for molecules like that are sort of on the edge. Really big molecules, charged molecules, are not going to be able to cross regardless of the fluidity or the permeability of the membrane. They're just never going to make it. But some molecules, it's sort of like, maybe. And for those ones, higher fluidity, they'll have a better chance to get across if the membrane is more fluid and therefore more permeable. Okay, I know this video is kind of on the longer side. We're getting back to videos with a lengthy one, so I apologize for that. But I hope this was useful, and we'll continue talking about membranes and getting across membranes in the next couple days. So I'm looking forward to seeing you guys soon. Bye-bye.